Welcome to Pluto. Pluto is the modern multiomics platform for translational discovery. Our software is a command center for translational scientists to manage their experimental data, discovery lists, prioritized targets and biomarkers, organize and manage all of their projects and programs, and even perform in-depth literature research. Pluto makes it easy to upload new data from an experiment, put that into a project, and run it through one of a variety of pipelines, including multiomics. Experiments can easily be viewed by their type, such as single-cell RNA-seq. Experiments can also be queried by organism and other attributes. A dataset in Pluto is very easy to navigate. Here you can see a single-cell RNA-seq experiment related to pediatric high-grade glioma. Each experiment contains an interactive notebook where scientists can include notes and protocols and other information about the study. Sample annotations help define where each sample came from. In this case, these came from different patients, and raw data was uploaded in the form of FASTQ files. Those raw sequencing files can be uploaded directly to Pluto, where they're stored securely in our cloud storage. Raw sequencing files can then be run through a pipeline which performs detailed QC, letting you know if the samples are in good shape to analyze. Looking a little more in depth at the analysis process, after the pipeline performing initial QC has been run, a scientist can then perform pre-processing steps, such as filtering, normalization, integration, clustering. All of this helps prepare your data for downstream analysis. In Pluto, all of this pre-processing and analysis is done completely without needing to code. There's a very intuitive interface for scientists, as well as instructions and tips, and detailed method sections informing the scientist exactly which underlying functions were used, and these are ready to go for publication, so they can be copied and pasted into your paper. Additionally, Pluto makes collaboration between scientists very straightforward, where you can @tag members of your team to ask them questions about your analysis. After performing clustering and refining your final clusters for single cell, you can move on to the annotation step. In annotation, any of the resolutions that you've decided to move forward with can be labeled. So here, for my 0.1 resolution, it can determine which of these cells are macrophages or tumor OPCs, and so on. This annotation can be done using AI models or biological domain expertise by looking at marker genes. In reality, cell type annotation is usually done with a combination of these. Lastly, downstream analysis in Pluto is also intuitive and requires no coding whatsoever. Here you can see an experiment that's already been analyzed in a variety of ways. These plots can all be edited and customized. Here we see a volcano plot where I can label and customize and highlight genes of interest, even lighting up certain pathways, like the diabetes pathway I might be interested in, or change colors. Similar to the pipelines, each individual analysis has its own method section as well, making it very easy for publication. Once a graph has been created, you'll of course want to export it for publication. With our export experience, you can customize the size and layout of a graph before exporting. So for this volcano plot, I can make it wide format, move that legend around, or make it tall, maybe put the legend in a different location. Get all of that close to publication ready, and then download in a variety of formats. In addition to experimental data analysis, Pluto can be used for a number of other applications, including analyzing published datasets. We have over 14,000 published experiments coming from GEO and other open repositories. Researchers can also directly import data from GEO and browse curated lists of pathways from MSigDB, such as looking at the hallmarks collection. Additionally, scientists can manage their own custom lists, such as targets, like high-priority targets for a therapeutic program that I'm working on. These target lists can be used to track the status of different targets and take detailed notes about those targets, including scientific evidence, reports that your team is creating, notes and comments between the team, as well as interactive evidence, such as literature or plots. Literature is managed in Pluto under Literature Collections, where I can create detailed literature collections pertaining to a topic of interest, such as Alzheimer's. Inside of these collections, I can then search for genes or topics and label those papers with custom labels. When it comes time to publish, you can send your data directly to GEO straight from Pluto, so there's no uploading or downloading needed. And all of your data is stored securely in the platform and can be easily filtered and searched, so you'll always know where to find your files. Pluto's comprehensive platform allows extensive integration with your own in-house apps or custom analyses, and we're always shipping new features in the platform. Overall, Pluto is a comprehensive multi-omics platform specifically designed with drug discovery in mind.